Neoplasia. Worldwide estimates of cancer cases are 18.1 million new cancer cases and 9.6 million cancer deaths annually. At least 42% of cancer cases in the United States may be preventable with lifestyle changes like smoking cessation, weight loss, physical activity, alcohol use reduction or avoidance, improved nutrition, the use of sunblock, and avoiding tanning devices. Vaccinations or antibiotic use can reduce the incidence of cancer-causing infections like those that are due to hepatitis B and C, viruses, the human papillovirus, and the Helicobacter pylori. Most cases of cancer arise sporadically from the accumulation of changes in DNA that may be influenced by environmental interactions. This feature is highlighted by the fact that overall cancer incidence increases with age, as uncontrolled cellular growth is the central flaw in a cancer cell. In multicellular organisms, coordinated cell division gives rise to tissues and organs during embryogenesis that are subsequently maintained by a balance between cell growth and cell death. The steps of the cell cycle must proceed in a careful, regulated manner to ensure the proper production of new cells. In its simplest categorization, the cell cycle is divided into two broad stages. Interphase describes the period in which the cell grows, replicates its DNA, and activates factors necessary for cell division. The M phase, or mitotic phase, entails the separation of chromosomes and cytoplasm. Interphase is characterized by three subphases referred to as G1, which is the first gap phase, S is synthesis, and G2 is the second gap phase. Cells with high rates of division will spend less time in G1 than cells with less frequent division. Throughout G1, high RNA and protein synthesis rates will support cell growth. During this time, cells make a commitment to completing the remaining steps of cell division. S phase is where the primary function involves DNA synthesis within the nucleus. During this time, the DNA from all 46 chromosomes is replicated, with each new copy remaining linked to the original by cohesive proteins. These connected chromosomes are referred to as sister chromatids. Production of histone proteins increases during S phase, and the DNA becomes tightly coiled around these histone proteins. This creates a DNA histone complex known as chromatin, which helps organize, condense, and package DNA in later stages of cell cycle. G2 is the integrity of the DNA is checked for errors and corrected if necessary by the DNA repair pathways. Following the completion of G2, the cell has reached a critical size, doubled its internal contents, replicated and checked its DNA, and it is now ready to divide. The process of mitosis involves condensation of DNA into chromosomes during prophase, while centromeres move toward opposite ends of the nucleus. Complexes attach to centromere regions of chromosomes in prophase, while the nuclear membrane begins to break down, allowing for binding of microtubules. Chromosomes are lined up during metaphase and separated by shortening microtubules in anaphase. The nuclear membranes form around newly separated chromosomes, which begin to decondense during telophase. During cytokinesis, an actin and myosin ring, a line at the center of the dividing cell, begins to contract. This serves to separate the cellular components and seal off the membranes of each new cell. CDKs are a family of kinase proteins that, when active, phosphorylate a specific set of protein substrates. The activity of the CDKs is regulated by their own phosphorylation and depends on the binding of proteins known as cyclins, which will increase the CDK function. A key feature of these interactions is that while cellular levels of CDKs remain relatively constant, the levels of cyclins rise and fall as the cell cycle progresses. Therefore, cell cycle regulation is controlled in part by fluctuations in the amount and the type of cyclins that is present. Of equal importance to cell cycle progression is the ability to halt the cycle when necessary. If a cell experiences alterations to its DNA through damage or mutations, the cell must be prevented from proceeding through cellular division so not to produce daughter cells with the same genetic alterations. The levels of cyclins vary throughout the cell cycle. While the cellular levels of cyclin-dependent kinases stay relatively constant throughout the cell cycle, levels of their activating proteins, the cyclins, fluctuate with the cell cycle phases. Along with additional activating mechanisms, these fluctuations determine the timing of transitions between the phases, controlling the rate of cell division. Progression through a G1 checkpoint, also known as the restriction point, ensures the presence of necessary growth conditions and commits the cell to the remainder of the division process. Checkpoint ensures that nutrients and growth factors are present and the extracellular environment is favorable for cell division. The TP53 gene encodes the protein p53, which is critical to halting the cell cycle when DNA is damaged or the cell has suffered injury. The p53 protein enters the nucleus and functions as a transcription factor, inducing the expression of numerous genes capable of halting the cell cycle, activating DNA repair pathways, or inducing cell death if defects cannot be reversed. Tumor is defined as an abnormal mass of tissue that results when cells divide more rapidly than they should and/or do not die when they should. A neoplasm refers to a new and uncontrolled proliferation of cells that can either be benign, which is non-cancerous, or malignant, which is cancerous. The cell terminology for cancer is based on the tissue origin. Proto-oncogenes are when some genes possess the ability to promote cellular proliferation and survival, but are subject to careful regulation to limit these functions to appropriate circumstances. Oncogenes will lead to sustained activity of their encoded proteins, resulting in unchecked growth that may progress to neoplasia. Tumor suppression is when many protective genes encode proteins that inhibit uncontrolled cell division and conduct surveillance of DNA. A proto-oncogene within a normal cell is mutated, becoming an oncogene. The oncogene product stimulates excessive and unregulated cell division. A tumor suppressor gene is inactivated by a mutation. The remaining normal allele sustains cell protection from proliferation. However, if the second allele develops an inactive mutation, tumor suppressor function and this pathway of protection from malignancy are lost. Hallmarks of cancer. Some of these characteristics will include uncontrolled proliferative signaling, evading growth suppressors, genomic instability, enabling cell immortality, resisting cell death, hijacking or generating blood supply sources for nourishment, and acquiring invasive and metastatic abilities. The RAS is an important intracellular signaling protein that, when properly stimulated, transmits signals that activate cell growth for a brief time before being turned off. A mutated RAS results in continuous stimulation that fuels tumor growth. When a tumor suppressor is inactivated or turned off, the likelihood of cancer development is now increased. When the P53 protein is not functional, DNA damage goes unrepaired and mutations accumulate in cells. This leads to acquisition of other cancer cell characteristics. A hallmark of cancer cells is the ability to bypass senescence and continue cell division. For tumors to continue to grow, they must develop an angiogenic ability to generate new blood vessels or expand the existing vascular tree. In order to metastasize, cells must separate from the original tumor, invade the surrounding tissues, enter and survive in circulation, lymphatics, or peritoneal space, and settle in a distant target organ where they now adapt, survive, and proliferate. A tumor is made of cancer stem cells, cancer cells, stroma cells, lymphocytes, macrophages, dendritic cells, and cells in some phase of the EMT. The tumor region closest to a blood vessel has normal oxygen supply and has cells that may maintain normal sensitivity to killing by the CLTs. The tumor region at a distance from the blood vessel has a hypoxic and acidotic environment that introduces cell stress. this may promote acquisition of cancer cell immune resistance and development of immune tolerance, as well as favoring mutations that will enable invasion and metastasis. Chief among the manifestations of cancer are inflammation with elevated cytokine production that may cause fevers and suppress appetite, while also promoting clotting. Hypercoagulability is also common and accompanies cancer, basically in the early stages. Tumor lysis syndrome is an acute generalized reaction to massive cell death caused by cancer treatment. The cells release their contents, causing hypouricemia, hyperkalemia, hyperphosphatemia, and hypocalcemia. The electrolyte imbalance that is caused will initiate an instability of excitable tissues, resulting in cardiac arrhythmias and neurologic seizures. Acute renal failure can also be a potential outcome. Germline mutations are mutations that occur in the DNA of a germ cell, such as an ova and spotozoa. Somatic mutations are mutations that occur in the cell spontaneously or as a result of mutagen exposures. Various environmental and occupational exposures can be part of the risk factors associated with cancer. This is hereditary cancer syndromes and their biology. These are various syndromes that are associated due to changes in chromosomes. Various viruses can also be precancerous. Genotyping in cancer helps to identify genetic mutations. Cancer in children is different from that in adults because it is not often of the epithelial origin, and it cannot be explained by environmental exposure. A primary malignancy of bone marrow, leukemia, results from the normal bone marrow components being supplanted by abnormal white blood cells. Osteosarcoma is an aggressive bone tumor affecting the long bones near the metaphyseal plate. Ewing sarcoma is a small, round cell, undifferentiated tumor that is believed to be of neurocrest origin. These are various signs and symptoms that can be seen in children that present with cancer. Four processes of aging that may promote cancer development are a favorable environment for cancer, an accumulation of cellular mutations, a decline in immune function, and alterations in hematopoietic stem cells. Increased cancer incidence with aging can be related to a number of changes across the lifespan. There are potentially multiple sporadic DNA damaging exposures, free radicals, carcinogens, and viruses, leading to altered expression of oncogenes, tumor suppressor genes, apoptosis genes, initiated of altered phenotype with increased cell proliferation, accumulation of additional gene alterations, promotion of the transition to malignancy with gradual acquisition of invasive and metastatic capacity, and tumor expansion later supported by angiogenesis. All of these are occurring while there is declining resistance in the host. When factors favoring malignancy outweigh factors that inhibit malignancy, this is when cancer can develop.